Can you offer any more on what that lyophilization at room temperature paradigm that you mentioned looks like? And then related question from Sogra, what do you think of stabilizing the mRNA in films? I'm assuming that's related to TFF.
Maybe first comment on the TFF, because you referred to the thin film freezing process. It's a tangential flow filtration we do as TFF downstream processing.
All right.
But just to sort that out.
Well, I was thinking
about that's what we do. Yeah, because thin film freezing is
I went right to thin film freezing.
Okay. Yeah. So what we developed is a lyophilization process that, and maybe to frame that a bit, the two largest problems we identified with regards to lyophilization is loss of integrity of the LNPs. Again, the two main aspects is agglomeration of the LNPs so that you get large aggregates that are undesired, and the other aspect is that the LNP falls apart, the mRNA is not in the LNP anymore and thus not active anymore. We have worked a lot on the lyophilization process because we expected that this would give a lot of benefit with regards to storage stability and long-term storage stability, and invested a lot of time on these processes. At the end, we developed a process on a pilot scale lyophilizer that allowed in lyophilization in a classical 2-hour glass vial, and then store the material and get a really classical lyophilization cake out of that. We can store it at different conditions. We know for 2 to 8 degrees, we have already data on more than 12 months, and at room temperature, as the studies are running, it's 6 months. But what we see is that we do not have any integrity loss during the process, meaning when we reconstitute the material, we have 100% intact LNP that is a 100% overlay of the LNP prior to lyophilization, and this maintains over time when we store the lyophilized cake. It's at the end, it's tweaking a lot on the different steps, and you have to identify what are the critical parameters to be addressed. But if you know them, it's in our opinion quite easy to tackle the problems and then get a valid process out of that. But saying this, I mentioned already before, it's a process that is controlled in a way that it should be easy to be translated into a large plant, but currently we are in the process of transferring that.
Yeah, maybe to allude a little bit more on this, it's clear that water is one of the main foes for stability of a messenger RNA strand. Yeah, you have the hydrolyzation mechanism where you have an attack on the 2' OH, and then you can induce strand breaks. So usually, if you are looking on long-term stability, you would usually want to maybe avoid that your messenger RNA is exposed to a lot of water. That can be done by lyophilization. Also, we were looking into this. I think we published a paper 8 years ago on this for the protamine formulation already. So that is a viable way to go. But also on your formulation, there can also be some aspects that you might manipulate to enhance the stability from the get-go. So the delivery system clearly plays also a role.
Yeah, absolutely.
Yeah. Sogra, hopefully those responses address your question. She did clarify in the chat box, by the way, that she meant thin film stabilization by using novel excipients. So naturally, you can see why I went to thin film freezing as opposed to tangential flow filtration. Miles, let's take a look at the results from that last polling question. So this is challenges, specific steps that are most challenging, and it looks like mRNA purification is far and away the the big one, followed by final formulation, template, DNA design and preparation. No surprises there, Christian?
No, I would have expected again this pattern, because DNA design and preparation is their methods available, very well known already. Also in the field, there are several CDMOs that deliver very high quality plasmid DNA. Linearization adds a level to that process, but I think that can still be controlled pretty well. The mRNA purification is, I think, more a new topic, and as mentioned, there's a lot of development ongoing there with the emerging field. There are several processes out, and a lot of development is ongoing. I think it's a great option to develop new processes that are different from what's known in the protein field and also what's known in other therapeutic areas. But I think it's a challenge, but it's already addressed very well. Not saying that there are not further opportunities to improve on that.
Yeah, also for me, it's not surprising how this comes up. Yeah, DNA technology is quite advanced in the market. It's well established. Yeah, you can buy it everywhere, more or less. Clearly, template design, you need to have some expertise there, but I think you can also apply this from even the vendors that produce you the DNA. The RNA purification, clearly, if you make mistakes there, you can have interesting data coming out. I think that was also the main obstacle for the whole mRNA field, that a lot of people, when they were doing messenger RNA-based research, were doing just in vitro transcription, then precipitation, and then use the messenger RNA for cell transfection. And in this case, you have a lot of impurities that will kill your cells, actually. And often people were then just seeing, oh, I do not see any transfection efficiency or translation, so I have degraded my RNA and I need to do more RNase protection. And I think that was a main obstacle in the past, yeah. And if you overcome this, you can actually work with this molecule very well. And I think in the future, it's always that people come up with solutions there. If you have an obstacle, you work on this and you crack the nut, and then you will have a process that might be even better than the existing ones that are on the market. We have, I think, zillion people now working on messenger RNA-based therapeutics. And I'm really looking forward to all these new technologies that will also help us to get better products for the patients.
Yeah. Excellent. Thanks, guys. So we are just about out of time. I've got one more question. Patrick, yeah, it was a beautiful segue, because you mentioned that you're looking forward to some of the new technologies that are coming out. And the last question I have for you was an audience question from Cedric that is sort of forward-looking, and it's related to quality. So as the field matures and becomes more common, and RNA becomes more commonplace, how do you think the QA/QC practices will change or improve? Thoughts on that? Like, what do you see happening in forward-looking statements around QA and QC and RNA?
Mm-hmm. I think on QC, we will get more and more understanding really of the really critical quality attributes, and also the QTPP. So on the quality attributes that you have to monitor in order to ensure efficacy and safety of your final product. And I think currently we are still struggling a little bit. A lot of different analyses are done. We sometimes do not have complete clear understanding what is critical and what not. And I think in the future, when this evolves more and more, the quality aspects will become more and more easy. From a platform idea, messenger RNA is very similar from the physicochemical properties. Yeah, usually it behaves like a large negatively charged polymer. and the process is usually also very similar to each other for each production. So I think driving a lot of different products within the similar processes and maybe even similar QCs. Some QC aspects will be protein specific or product specific, but other ones will be platform specific, and then you can leverage a lot. I think that will also have then aspects on QA, if this is answering your question.
Thanks, Patrick. Christian, anything to close out with?
I think to add on that, there's also a lot of happening on the markets and the manufacturers of kits of assays for analytics to be used also in early stage of development on specific resins for analytical columns to better analyze your LNPs and your mRNA, as well as device manufacturers that simplify the analysis of your drugs, and I'm looking forward to what's coming up in the next few years on that.
Perfect. Well, like I said,
Patrick,
yes.
Sorry. We also have to highlight here the role of the regulators, and I think here that's
definitely
really the prime time. They will see all the data, and they will come up then also with guidances how you can do and develop safe and efficient drugs. And I think it's not a burden or an obstacle that they have now understanding, that they look into this and that they give you advice and that they are demanding things from you. It's really helping you. So you should really hear to the regulators and work with them.
Yeah, it's a great point, Patrick. And I think the regulatory considerations would make a whole nother hour-long presentation in themselves, especially as those play out here. So that hour went by fast, didn't it?
Yes.
It did indeed.
I appreciate you two for subjecting yourselves to my questions and those of our audience. I appreciate our audience for coming out and asking such great questions. And we got a bunch of them. And here's the good news. Even though we didn't get to all of the audience questions, the good news is that next month in October, you'll be receiving an invitation to another Bioprocess Online Live event, also focused on mRNA therapeutic progress. We're going to welcome Omega Therapeutics Executive Director of Process Development, Analytical Sciences, CMC, and Tech Ops, Dr. Jeffrey Atkinson, into that event, as well as Combined Therapeutics Chief Scientific Officer, Dr. Thomas Van Cott. So we'll get you an invitation to that. I believe it's the second Tuesday in October. Stay tuned. In the meantime, please do check out the resources on your screen, displayed on your screen. We got the latest Bioprocess Online guest columns up there on mRNA development, technical resources from Roche. We've got the Business of Biotech podcast. Thanks again to Roche for sponsoring this, by the way. And remember, within 24 hours, this presentation will be available on demand using the same link you used today to join us. Be sure to share it with your colleagues. I'm Matt Pillar. This has been the Bioprocess Online Live. Patrick and Christian, thanks again for joining me.